Well, hopefully you guys can, oh, good, somebody's logging in. Sorry for the delay in getting started, but actually there was no Zoom meeting set for today, so I had to create one, and I tried to create it with the same information so you guys could just log in. I'm not sure. I must have set up the meetings wrong. Are there any questions before we begin?
Nope.
Thank you. Scrolling down. Have we finished chapter 7?
Yeah, we finished on
It's finished. Great. All
Thursday.
right. So all I need to do then is a review. I actually should have done that review in the lab, I guess, because you're having the final on the 13th. All right, let's begin the review. Any questions? Let me share my screen. What I'm going to just do is mention that the exam, let me call up my schedule here. The exam will be heavy, about 50% of the questions from the material that you have not been quizzed on. Our last quiz covered chapter 8 and lab 9. So that means there will be about 50% of the material on chapter 14, chapter 13, and a few questions on chapter 7, but not a lot on chapter 7. And then there will be questions on the unknown project. Any question about what 50% of the material will be on? Now this says lab module 9 for the heavy coverage of the final. So let's add lab 9 as well. It looks like I added lab 9 to chapter 5 quiz, but just study lab 9 also. Now the rest of the chapters, there will only be one or at most two questions from each chapter, but the final is comprehensive. So don't ignore what you previously learned, but don't put the majority of your study effort in the other chapters because, like I said, there will be only one or about two questions from all of the other lessons. There is one exception, and that is chapter 1. Chapter 1 introduced many topics to you that we then went on and later talked about in greater detail. So you will get more questions from chapter 1. Also, chapter 1 covers the history of microbiology. And if you haven't learned, I really enjoy the history of microbiology. So I will ask more questions on chapter 1 than I will on other chapters like chapter 2 and 3. Any question about any of that?
Will there be more questions regarding past labs?
What do you mean? Chapter lab like
Just
0
like,
and 0, lab 1, things like that?
yeah, or is it more so just like chapter review?
There will be some questions from the labs, but there will be no more than one question from each of the earlier labs, with the possible exception of lab 9. And I think lab 9 only might have like two questions on it. So yes, you should not just forget about your previous labs. you should lightly review the earlier labs, like lab 00 to lab 8. You should just lightly review them if you have the time. The majority of the lab questions, of course, will be on the unknown project. There are no questions about the infectious disease project because each of you had a different topic. But do understand the unknown project, and if I give you information about a bacteria, then you're going to be expected to solve it. And I will give you a key or something like that to solve it. I think that's about it for the type of questions. And like I said, the majority of the questions will be chapter 13 and 14. Only a few from chapter 7. And then there's definitely a few from chapter 1. And then only about one from every other, or one or two from every other chapter. So let's begin chapter 7. I'll just start backwards because the majority of the questions will be on the last lessons. On chapter 7, you should know the terms like sterilization, disinfection, antisepsis, etc. You should be able to discuss the factors that influence microbial death rates. And I don't want to go into that in any great detail, but I've got it mainly on one slide here. These factors here. Be able to discuss the three main ways in which antimicrobial agents kill or inhibit cells. One, they damage proteins, or what's the word? Drawing a blank here. When the protein folds out of shape, we call it a specific term, and whatever that term.
Denature?
Denature, thank you, denature proteins. The second is to damage cell membranes, like put holes in them. Polymyxin B is an antibiotic that works in that way. And there's other agents like alcohol, which can do that. It can damage cells by putting damaging the cell membrane, although alcohol can also denature proteins like enzymes. And then there's damaged DNA, and like ionizing radiation or even UV light damages DNA. So those are the three ways that antimicrobial agents kill or inhibit cells. You should understand the various types of disinfectants and how they work, and generally just know a little bit about that. I think I've got a slide on that. Let's see if I can get that. Blow this slide up. So you should know the different categories of agents: moist heat or dry heat, filtration, cold, which really doesn't kill cells, even when you freeze them and then thaw them out. High pressure, desiccation, osmotic pressure, radiation. Those are physical methods. Let's move on to chemical methods. You should know the phenol and the phenolics. I don't think you need to know the biguanides, but you should know the halogens, the alcohols, the heavy metals. That would be something like silver, copper. What's another heavy metal? Sulfate, other heavy metals. Surface active agents. These are mostly soaps and detergents, although the hospital uses quaternary ammonium compounds. Then chemical food preservatives like nitrates and nitrites. I don't think I asked about the aldehydes. You should know about the ethylene oxide. It's a very toxic gas and it's very poisonous to people, but it's very good in the sense that you can kill endospores with it, but it takes a long time exposure, at least to kill endospores. And then peroxygen, like hydrogen peroxide, which you can find in every house, is actually a very good sterilizing agent, and it does kill endospores. Any question about any of that? I suppose you should know this slide too, and that is, let me back, get this out of the way. Ah, come on, there it goes. Soap and water, not very good. Disinfectant or antiseptic. Nobody washes their hands for 2 minutes. And when you do, you only kill about 10% of the cells. Tincture of iodine, which is the iodine most of us have in our house, a very good disinfectant. And it used to be used in the hospital before surgery. Now they have better agents, but they used to use this all the time. And you just put it on the patient when you're going to have where you're going to have the surgery. And it's long-term exposure. And then it does the good idea that it coats the area that's affected. So the surgeon knows this area is sterilized. And anyways, it doesn't not really sterilize, but it's been treated and a very good treatment. All right, let me go back to where I was in the show. Talked about 4, so understand the relative resistance of the major microbial groups to antimicrobial agents. And that's the 3 last slides. Understand this hierarchy. Prions are the most difficult microbe to remove. And to tell you the truth, we haven't quite figured out entirely how to remove prions, but you can incinerate them, and you can also treat them with proteinases. Endospores, very difficult to remove. There are only a few antimicrobial agents that do kill endospores. Mycobacteria, difficult to kill because of the macolic acid in their cell walls. If you treat with a chemical agent that is water soluble, it's probably not going to affect the mycobacteria. And then all the way down to viruses with lipid envelopes, the easiest thing to kill or inactivate, and that's because you can damage either the nucleic acid or the lipid of the virus, the envelope, and then damage the virus. It's no longer effective. And you should realize that each of these two have different characteristics, like alcohol, which is a very poor antiseptic, that we call it a degerming agent. It's so poor of an antiseptic that it actually is very effective on mycobacteria because the alcohol gets right through the macolic acid of the cell wall in the mycobacteria. Any question about any of that? All right, that's it for chapter 7. Let's move on to chapter 13. Make it chapter 14, sorry. In this chapter, you should know the terms, because if you learn the terms of this chapter, you'll learn 50% of the material of this chapter. I've only got a few terms here, but know all of them. Understand the relationship between the normal microbiota and its host. We have a normal microbiota which lives with all of us, and it is mostly on our skin, our mucous membranes. The normal microbiota is not usually internal to the body other than in the mucous membranes, which go inside, like the gastrointestinal tract is coated with the normal microbiota. The lower urinary reproductive tract is also coated with the normal microbiota. Understand the stages of disease development. All diseases go through a typical stage and development before disease develops. Understand what nosocomial infections are and how to control them. And this is really one of the most important topics that you guys can learn in microbiology, because if you go out and work in the healthcare facility, you have to work to prevent nosocomial infections. These are infections a patient gets from staying in a hospital or a hospital-like setting. And they would not have gotten that nosocomial infection if they had not gotten in the hospital-like setting. This is a major concern in America as well as the world, and it's also a major killer of patients, mainly because the patient is already weakened, and then when they get a nosocomial infection, it could be enough to break the camel's back, or whatever that expression is. That it just puts them over, and then they're unable to continue living. Any question about any of that? Let's talk a little bit about the stages of disease development. See if I can find that. Oh, let me state the normal locations of the normal microbiota only on the outside of the body and the internal mucous membranes. There is a difference between the normal microbiota, which colonize the host generally for about the lifetime of the host. And that's different from a transient microbiota, which may be present for a short time. And that there's more normal microbiota than there are human body cells. And I have to congratulate the student who answered this question, and they said, although there's more normal microbiota, the volume of the human body cells is greater than the volume of the normal microbiota. And of course, that's true because the body cells are about 10 times the size of the bacteria cells. And I'd never seen that comparison before, so I congratulate the student who came up with that number. You should know there's some unusual places where we found the normal microbiota, like mother's breast milk and the female bladder. I've looked and I could not find any research on the male bladder. And so we'll just go and say female bladder. Oh, you should know about Koch's postulates. We've talked about that 3 different times in this class. So I will be asking a question on Koch's postulate. This is the steps one uses to prove that a given agent causes a given disease. Now, you should realize that there are exceptions to Koch's postulates, such as when you cannot grow a organism in tissue culture, like we've never been able to grow syphilis or Bartonella henselae, the organism which causes cat scratch fever in tissue culture. We just don't know what the cells require to grow, and we've never been able to figure out how to get them to grow in tissue culture. And people have been trying to grow these organisms for over 100 years now. So there are exceptions where you can't use Koch's postulates. And then when there's moral and ethical conditions for human-only diseases, like nobody's ever injected HIV in patients to determine that HIV causes AIDS. And what you use is you simply use massive epidemiological studies and then follow these patients over time to serve the same function as Koch's postulate will serve, meaning to show which organism is causing a disease. Looking for the different, well, we should know about latent disease and herd immunity. You should know about the patterns of disease. All disease comes from a reservoir. The pathogen has to be transmitted to a susceptible host. The pathogen has to multiply, and the pathogen has to injure the host. Without injury, there is no signs or symptoms. And then when there's injury, the patient comes down with disease. Now, the patient can harbor the host, and the disease organism can be asymptomatic. The patient doesn't know they have disease, and they can spread it to other people. But for them, they don't even know they have disease because the pathogen is not injuring the host. But it may be growing in the host. You should know about the predisposing factors of disease. Here we go, the development of disease. You should know all disease has an incubation period, the time before, the time when the organism first enters the host, and the time before the patient has any signs or symptoms of disease. That's followed by the prodromal period, when the patient has very mild signs and symptoms. Sorry here, I'm trying to blow my nose out of sight. The prodromal period ends when the patient has no longer mild signs or symptoms, they're severe. And this is the period of illness. The period of illness continues until the patient starts getting better. Like when a patient has a fever, once the fever breaks, the patient, we say, is getting better. That starts the period of decline. The period of decline continues until the number of organisms in the patient drops below a threshold number where the patient no longer has signs or symptoms of disease, but they still have the organisms, and this we call the period of convalescence. And the period of convalescence ends when the patient no longer has any organisms in the patient. So they can spread disease at any period of disease, although they're much less likely to spread it in the period of convalescence. And they're actually also not likely to spread it early in the incubation period. But with some common diseases like colds and flus, at least for adults, they're most likely to spread it early in the late in the incubation period or in the prodromal period. In the period of illness in an adult, the immune system is ramping up, and so the patient is less likely to spread the disease while they are an adult. And my guess is that they're trying to prevent spreading the disease to their offspring. Any question about any of that? All right, so most diseases follow this stage. The only exception is if a stage is missing. Like sometimes I'm walking around, everything's fine, and then all of a sudden it feels like somebody's hit me over the head with a sledgehammer. That's usually what happens when I get the flu. There is no prodromal period. And then there's some diseases where there's no period of decline or period of convalescence, and that's usually when the disease kills the patient. Any questions about any of that? We should know the transmission of disease, contact, vehicle, and vector transmission. And then, of course, you should know about nosocomial infections. Any question about that? All right, if not, let's move on. Chapter 13. viruses, viroids, and prions. You should know the terms of this chapter, understand the characteristics of viruses, the structure of viruses, and the classification of viruses. And know that there is no Latin names for viruses. We simply use common English names like polio virus. That's actually the scientific name for the virus. You should understand about viral multiplication and the viral life cycles. With the bacteriophages, there's two life cycles. The lytic life cycle, where the virus will grow within the cell, multiply, and then lyse the cell, releasing the viruses. And then there's the lysogenic life cycle, where the virus will infect the host cell, and the virus has to decide immediately once it infects the cell whether they enter the lytic life cycle or enter the lysogenic life cycle. If it enters the lysogenic life cycle, it integrates into the host chromosome of the cell, and then the only time the virus multiplies when it's in the lysogenic life cycle is when the host cell divides. It'll multiply its DNA and split that DNA into two cells, and that DNA will include the viral DNA integrated into the host chromosome. Any question about that? Animal viruses do have the lytic life cycle. They do not have the lysogenic life cycle, but they have something similar. They have the budding life cycle or a persistent infection life cycle, where the virus generally multiplies at low numbers in the cell. And if it follows the budding life cycle, the virus will bud out of either the cell membrane, a nuclear membrane, or the endoplasmic reticulum membrane to obtain the envelope of the virus. The persistent infection, I probably won't ask any questions on, but it's similar to the budding virus, and that is not large numbers of viruses are made. And when the virus is replicating, the virus is put in a vesicle, which will then travel to the cell membrane and fuse with it, opening it up so the contents within the vesicle can then be dumped out of the cell. The persistent viruses are naked viruses, and then the budding viruses are envelope viruses. Any question about that? Animal viruses, or I should say, no, it is animal viruses. Animal viruses also have the latent life cycle. This is usually a budding virus, which goes latent. When the virus is latent, it's no longer reproducing. It's really not causing disease, and it's in stasis. It's not even really metabolically active, or if it is, it's at low metabolism. The virus just hiding out, waiting for an opportunity to leave the latent life cycle, and then it'll re-enter the budding life cycle, and the virus will begin multiplying. Two examples of that is the child comes down with chickenpox, and then usually decades later, the chickenpox virus can reactivate, leave latency, and enter the budding life cycle. And then the child, who's usually an elderly adult at this time, gets the shingles. Another example is the herpes simplex virus type 1, which causes cold sores. When you have a cold sore, the virus is in the budding life cycle. And then when it's not, you don't have a cold sore, the virus is in the latent life cycle. And it's just hiding out, waiting to leave latency and enter the budding life cycle and reproduce again. Any questions about any of that? All right, lastly, you should understand what prions are. They are proteinaceous infectious particles, which actually reproduce in a way. And what happens is the prion comes up to the normal cellular protein, and the two have the same amino acid sequence. And then the prion protein causes the normal protein to change its folding pattern and become a prion protein. The prion proteins then multiply in the cell because the nerve cell knows it needs more of the normal protein, and so it makes more of it. And more prion protein gets made, and that will eventually form plaques in the brain and kill the cell. And then you should know about viroids. These are plant naked RNA molecules which cause disease in plants. Fortunately, there are no viroids, at least there's none known, in animal cells. So all the viroids only cause diseases in plants. Any question about chapter 13? Let me quickly go through and see if there's anything particular I want to. Oh, you should know about the host range of viruses. Most viruses are very specific about the cells that they infect. They usually only infect a few cells of one or two species, like HIV infects, let's see, macrophages. I think they infect nerve cells, brain cells, and they infect helper T cells. But there are some exceptions where a virus, like some of the flu viruses and some of the encephalitis viruses, do infect numerous species. Okay, so there are some exceptions, but most viruses are very specific in their host range, and they usually only infect one or a few species, and usually only a few cells in those few species, like HIV. You should know about the virus sizes. Yeah, you should know about the virus lytic life cycle following 5 stages: attachment, penetration, biosynthesis, maturation, and then release. Release by bursting the cell. You should know about the lysogenic life cycle. Oh, here's something. You should know the consequences of lysogeny. A cell that has a lysogenic virus in it will be immune to infection by that species of virus, and then that can give the cell new properties because the cell has new DNA, and that is called phage conversion, changing the potential properties of the cell. The best example is Corynebacterium diphtherium, only produces the Corynebacterium diphtherium toxin when the bacteria has the lysogenic virus in it. You should know animal viruses can multiply in the nucleus as well as the cytoplasm. Bacteriophages can only replicate in the cytoplasm. Well, you should know retroviruses are a little different than most other viruses because they have reverse transcriptase in them. And I could mention that there is some other virus. I think it's hepatitis B, which has reverse transcriptase in it. But generally, a reverse transcriptase is found in the retroviruses, and it allows replication of RNA to make DNA. And that does happen in the hepatitis B virus, but hepatitis B virus, I think, is a DNA virus to begin with. But it does have reverse transcriptase. You don't need to know that. Sorry I brought it up. You should know about the COVID-19 is a coronavirus. All right, I think that's it for this chapter. Any questions about chapter 13? If not, let's move on to chapter 8. Chapter 8 is a fairly important chapter, so I probably will be answering at least 2 questions on the final from this. You should know the terms of this chapter, be able to describe DNA replication, RNA transcription, and protein translation, and then understand the different types of mutation. There's a point mutation where simply a nucleotide base is changed. Trying to get there. I don't see it here. Well, I'll come back to that. So where a simple base is changed, this we call a point mutation or a base substitution, where one base is substituted for another base, which is the change in the DNA. And that results in 3 different types of mutation: a silent mutation, a missense mutation, or a nonsense mutation. I'll talk more about those in a minute. You can also have a different type of mutation, a frame shift mutation, where one base is added or one base is deleted, or some multiple of bases are added or deleted, as long as it's not a multiple of 3. That changes the frame shift reading of the codon. And so after this frame shift, the codons will be totally changed. And after that frame shift mutation, then you get a totally different protein, because each amino acid after that has a different codon. And that's a frame shift mutation. The simplest way is you simply add a base or you subtract a base, and then that changes the codon. Well, codons are read as 3, so the next one will be changed, and then the next one and the next one. Any questions about that? There's also a macro mutation, which is not discussed in your book. I don't know why your textbook does not change it or discuss it, but that's when the DNA is changed in a large region, where an entire region of a chromosome is either added or deleted. And it can include the entire chromosome, Down syndrome being an example, where everyone who has Down syndrome has an extra chromosome number 23. Another example of a macromutation are individuals with XXY phenotype, where they have an extra, well, either an X or a Y chromosome. Okay? The XXY individuals actually appear normal, and the XYY individuals also appear phenotypically normal. Any question about any of that? All right, let's talk a little bit about silent mutations. That's where you change the DNA, so the base is changed, but it results in the amino acid not being changed because the codon is changed, but that codon still can't codes for the same amino acid. So the protein is not changed, and that's why we call it a silent mutation. The DNA is changed, but the protein is not. A missense mutation is where you have a point mutation, the base is changed, and the amino acid sequence is changed. A missense mutation. So the protein has a different amino acid at this location. The best example is sickle cell anemia, where there's a point mutation and only one amino acid out of thousands of amino acids in the protein is altered, and it results in the sickle cell anemia phenotype, assuming you have 2 of those genes. And then there's a nonsense mutation, where you change the DNA base and it changes the codon to code for the stop codon. So what you do is you make a truncated protein instead of the full protein. Usually that inactivates the protein. Any question about any of that? All right, those are the three base substitution. I already talked about frameshift mutation and macromutation. I think that's all I want to talk about. You should know about genetic recombination. That's where there's an exchange between genes, and then there's crossing over, which is recombination, but it's more specific, where the chromosomes literally come together, cross over, and then switch parts. This normally happens in both mitosis and meiosis. I think that's right. And that we call crossing over. Recombination is a little more general, where the genes change. Doesn't have to involve crossing over, but it often does, but it can be more general. And then you should know about transformation, transconjugation, and transduction, how existing genes in a population can spread. And you should know about transposons. DNA in the genome that can move from one place in the genome to another place in the genome. All right, any questions about chapter 8? Chapter 7, which I should have done earlier because that one hasn't been reviewed. Know the terms of this chapter? I've already discussed this, haven't I? All right, let's move out of there. Chapter 6. Somebody could confirm that just to make me feel better. Know the
We did
terms.
cover 7.
Yeah, thank you. Know the terms of chapter 6. Some important terms: reproductive spores. They differ from endospores. A reproductive spore, one cell makes several spores. Usually that's like 4 or something like that. And then the parent cell survives. So it's like one cell makes 5 cells. That is reproduction. An endospore, one cell makes one endospore, and then the parent cell dies. So you get one cell to begin with, and you end up with one cell. That is not reproduction. Endospores are a survival strategy. And then endospores can survive almost anything you throw at them. They're very hardy. They live for a long time, just waiting for the environment to change, to become favorable for the cell to grow, in which case it will come out of the endospore and make the vegetative cell, and then the vegetative cell will become metabolically active and reproduce. You should know the terms like acidophiles, organisms that like acids, halophiles, organisms that like salt, aerotolerant anaerobes, organisms that are anaerobes. They do not need oxygen, but they can tolerate oxygen because they have all the enzymes and anti-metabolites to survive oxygen, and the oxygen metabolites, which are toxic to cells. Facultative anaerobes are organisms which are anaerobes, but they can reproduce aerobically, and they prefer to do that because it produces the most ATP, So the organisms grow best and fastest when they're growing aerobically, but they do not require oxygen to grow, and they can grow anaerobically. I should probably mention briefly that slide. Growing microbes in thioglycolate. There are aerobes that do require oxygen, and they only grow at the surface of the thioglycolate tube. There are the obligate anaerobes, which only grow at the bottom of the thioglycolate tube where there's no oxygen. There's the aerotolerant anaerobes, which grow everywhere evenly in the tube. The facultative anaerobes, let me blow this up, grow best at the top of the tube because they can grow fastest there. But they do grow everywhere in the tube. And then there's the microaerophiles. They are aerobes, so they do require oxygen to grow, but they grow best at reduced oxygen concentrations, usually between, what is it? Something like 2% oxygen to 10% oxygen, something like that. They grow slower at full oxygen, around 20% oxygen. So they grow best someplace in the middle of the thioglycolate tube, not at the bottom and not at the top. They do perhaps grow at the top, but at a reduced rate. All right, let me end this here. Go back to where I was talking about. Understand the various requirements for growth. All organisms require certain chemicals and physical environments to grow, and they must be in those to grow, meaning you can't grow cells in a vacuum like in space, and you can't grow cells in Antarctica where it's frozen. Although microbes do grow there, but I'm just saying normal cells can't grow when the temperature is below zero. Understand the four phases of growth, and then compare the different methods for measuring microbial growth. Any questions about chapter 6? Let's move on to chapter 5. Know the terms of this chapter. Know about enzymes, like what's an active site, what is a allosteric site. Know about competitive inhibition and allosteric inhibition, also called non-competitive inhibition. Know about redox or oxidation reduction reactions. Know what are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. Know about aerobic respiration, because this is what we mostly talked about in this chapter. Know about its steps and how it differs from anaerobic respiration. You should also know about fermentation. I'm surprised that's not on here. And then know about cyclic photosynthesis as well. I think that's it for this chapter. Obviously know the terms catabolism and anabolism. Know about enzymes. Know about the factors that can influence enzyme activity. All enzymes work at best at an optimal temperature, an optimal pH, an optimal salinity, meaning optimal salt concentration. And when you move them away from that optimum, the enzyme will start unfolding, will have less activity. And when you move it far away from its optimum, the enzyme will no longer work. And that we call a denatured enzyme or denatured protein. Now, I should know about ribozymes, enzymes which are made of RNA. I'm just going to say you should know about aerobic respiration and the summary of aerobic respiration. You don't need to know all of the details, like all of the different steps of aerobic respiration, but you should, like of glycolysis, you don't need to know all the steps of glycolysis, but you should know what you start with in glycolysis and what you end up with. So the summary equation. And you should know what you're starting with in the Krebs cycle and then what you end up with. And the same with the electron transport chain, where you start with when you start that step and what you end with. And then also what you start with aerobic respiration, glucose, oxygen, and then you end up with CO2, ATP, and water. All right, you should know the 3 ways of making ATP: substrate level ATP, where the phosphate is transferred from one molecule to ADP, making ATP. It actually happens in glycolysis. You don't need to know that, though. You should know the second way of making ATP, that's respiration. This can be aerobic or anaerobic respiration, where we have a sequential transfer of an electron moving down an electron transport chain, making ATP. And then photophosphorylation, happening only in photosynthetic organisms, where light energy can be converted to make ATP. In making ATP in photosynthesis, oftentimes that ATP is used up to make the glucose. So when we're talking about non-cyclic photosynthesis, the ATP is made, but it's used up to make glucose. In non-cyclic photosynthesis, in photophosphorylation, you change light energy directly into making ATP. All right, I've already talked about aerobic respiration. You should know how it differs from anaerobic respiration. And you should know about fermentation, where you make 2 ATP molecules per molecule of glucose. How you do that is in glycolysis. And then you follow the glycolysis steps with fermentation step or steps. There's usually 1 or 2 steps of fermentation to convert pyruvic acid into the fermentation end product. And we're only going to talk about 2 end products, alcohol and lactic acid, which happen in alcoholic or ethanolic fermentation or in lactic acid fermentation. Any question about any of that? Now, I should mention that other molecules can be used besides glucose. It's just organisms prefer to use glucose because it's easy to use and the pathway is set up for glucose. And generally speaking, for us couch potatoes, we'll change everything we eat into glucose and then send glucose down the normal pathway, which for us will be aerobic respiration. It doesn't have to happen that way. Like marathon runners can metabolize lipids, fats directly with aerobic respiration, but they do not do that until they're 45 minutes into their marathon run. So even they prefer to directly use glucose and then send it through aerobic respiration. And when you eat carbohydrates, all of the carbs will be converted into glucose and then sent through aerobic respiration that way. Some of them are easier to do than others, like starch is just broken down into glucose, and then you send glucose into the aerobic respiration. When you eat honey, the fructose has to be digested, taken to your liver, and then converted into glucose, and then the glucose is slowly given back to your blood. That's why diabetics can actually eat fructose. You should know about amphibolic pathways, the bridge between catabolism and anabolism. All right, that's it for chapter 5. Any questions? All right, know the terms of chapter 4, and you only need to know the anatomy of prokaryotes. We're not going to talk about the anatomy of eukaryotes. I'm certain I did not ask a question on that on the final. The only thing you need to know about eukaryotes is the endosymbiotic theory, the theory about how eukaryotes obtain a mitochondria for the animal cells, and then for plant cells, how they obtain the mitochondria and the chloroplast. Any question about any of that? And that is those proto-eukaryotes simply ate or consumed a bacteria, which they did not then digest, and they started living in symbiosis with it. And that bacteria eventually became the mitochondria in animal cells. And in plant cells, they did that to two bacteria. One became the mitochondria, and one became the chloroplast. You should know how gram-positive and gram-negative cell walls differ. There will probably be a question about that. And then describe the importance of taxus in the bacteria. So you should know the prokaryotes and eukaryotes are very different. So you should be able to say how they're different. I don't think I asked any question about the different shapes and sizes of cells, but you should know that. You should know about the glycocalyx. You should know about endoflagella. This is a flagellum that is actually inside the cell wall, as shown here. And it doesn't whip like a normal flagella. What it causes is the spirochete to have the corkscrew-like movement. So spirochetes have the endoflagella. You should know about the sex pili, which allows the F minus cell here to literally skewer the F plus cell, and then the F minus cell can give a plasmid that can move across the sex pilus into the F minus cell, and then that'll transform the F minus cell into an F plus cell. That plasmid actually encodes for the sex pili. So the F plus cell will become, the F minus cell will become an F plus cell once it gets that plasmid. You should know about the cell walls, gram-positive, gram-negative cells. You should know that mycoplasmas differ and they do not have a cell wall. They are a bacteria. And then you should know the Mycobacterium, also bacteria, differ in that they have a different cell wall. It has mycolic acid in it, giving the Mycobacteria different properties. Mainly, they are resistant to an agent which is water soluble, because water cannot easily get through the mycolic acid. So something like bleach, which is water soluble, is not very good at killing Mycobacterium. You know about selective permeability. All right, that's it. Any question about chapter 4? Let's move on to chapter 3. Know the terms of chapter 3, observing microorganisms. You should know about microscopes, understand the size of organisms and the properties of microscope, understand the difference between light microscopes, which use light, and electron microscopes, which use electrons. Electron microscope can magnify many times more than a light microscope. And we should mention that the light microscope can't see anything smaller than the, I think it's about half the wavelength of light. So it's limited by the properties of the wavelength of light. And that is what causes the I've got that in here someplace. Not finding it though. Limit of resolution. The ability of the lens to distinguish 2 points as 2 separate independent points. The resolving power of a microscope is 0.02 micrometers. The reason for this limit is white light cannot magnify beyond 0.2 micrometers because of the wavelength of white light. Any question about that? So that means the total magnification we can do, the best we can do, is about 2,000 times, because when you magnify further, you're magnifying beyond what light can resolve. And so it's empty magnification. So the best microscopes only magnify 2,000 times. And once again, that limit is based on the properties of light. Anything smaller than 0.2 micrometers cannot be distinguished by the light microscope because of the properties, the wavelength of white light. Any question about any of that? Now, in theory, if you used a certain wavelength, I don't remember if it's blue or red, you could go a little bit beyond this, but not much. And surprisingly, nobody's ever done that, made a microscope using, it's either blue light or red light, but it could resolve a little bit better than this, but not much better. You don't need to know that last part. I'm sorry I even brought it up. Resolution, you can resolve these 2 points because they're separated by 200 nanometers. That's 0.2 micrometers. When they're closer than 200 nanometers, you cannot resolve them. And these 2 points are 200 nanometers apart. You should know about refraction, the bending of light, when light moves from one media to another. such as when light's moving through water and then moves through air, it causes the bending of light, making it look like this pencil is bent. And that is just because of the refractive index of the medium that the light is moving through. Air has a different refractive index than water, and when that happens, light will refract or bend. And that's why we use oil, because oil has the same refractive index as glass, the glass of the slide, the glass of the ocular lens, and then the oil in between prevents the refraction of light. No light is bent. That means more light, this one here, will get collected by the objective lens, and then we can have a higher resolution because you're collecting more light, whereas this light ray will be refracted away from the lens, and so oil improves the resolution of the microscope when you use oil. Any question about any of that? All right. You should know the two major types of electron microscopy. Transmission electron microscopy, shown here, where you see an object in 2 dimensions. You can see within the cell, but only one plane of the cell. And then scanning electron microscopy, which only sees the surface of the cell. This shows the object in 3 dimensions, not 2 dimensions, but you can only see the surface of the cell. Any question about any of that? Or the surface of an object? Scanning electron microscopy and transmission electron microscopy. The other two you don't need to know about. There are 2 other forms of electron microscope. You should know why we use a dye to stain the cells. It's not to necessarily make the cell, I don't know, appear better. What it does is it increases the contrast between the cell and its environment, the background, so the cell just shows up better. So you increase the contrast whenever you use a dye or a stain. I should know the 3 different types of stains. A simple stain, where you're only using one stain, all the cells have the same color. A differential stain, which shows differences between cells. Usually, the cells have 2 different colors because you're using 2 different stains. That isn't required on a differential stain. For example, the differential stain could stain one cell and not stain another cell, or it could stain one cell very darkly and another cell only very lightly, but still a differential stain. But usually using 2 different stains, like in the Gram stain for the differential stain, and the Gram stain is the most commonly used differential stain. Special stains, there's a special stain that we use to stain a special part or organelle or something of a cell. So a special stain only stains a special character or property of the cell, like the flagellum stain is a special stain, staining the flagellum. You should know the properties of the Gram stain and the difference of the Gram-positive cell and the Gram-negative cell, and why the Gram stain stains the Gram-positive cells purple and the Gram-negative cells pink. All right. Any questions about chapter 3? If not, let's move on to chapter 2. Chapter 2 should have been mostly a review for you from Biology 160. You should know what organic and inorganic compounds are. Organic compounds can be hydrocarbons or another organic compound. Uh, with respect to the 4 biological molecules, be able to describe the relationship and the characteristics of the monomers and the polymers, which are found in, as far as I know, all carbohydrates, all proteins, all nucleic acids, but only some of the lipids have monomers and polymers. Some of the lipids, like the cholesterol, oh gosh, I'm dropping the name. Cholesterol is not made up of monomers. It is its own one large molecule. So some of the lipids have monomers, like the triglycerides and the fats made up of fatty acid and glycerol. Phospholipid also has monomers. One other lipid has monomers, but I'm dropping its name. But the waxes and the, what are those called? Cholesterol. Sterols. There we go. I got it. Didn't you have to look it up? The sterols uh, and steroids are, they're not made up of monomers. All right. So you should know the building block of carbohydrates are simple sugars. The building blocks of proteins are amino acids. The building block of nucleic acids are nucleotides. And then lipids are a little different, where some of them, like the steroids, the sterols, and the waxes, have no building blocks. They are their own molecule. But the fats, like triglycerides and the phospholipids, are made up of fatty acids, glycerol, and that's all it for the fats and the triglycerides. But for the phospholipids, they're made up of fatty acids, glycerol, and then the uh, phosphate group. Any question about the chapter 3? You should know about the 3 levels of protein structure in all proteins, and some proteins have 4 levels of protein structure. That will be a protein made up of more than one polypeptide, like an antibody made up of 4 polypeptides to make the antibody molecule, and it has a quaternary, 4th level of protein structure. All right, I think that's it for chapter 2. Let's go on to chapter 1. Chapter 1, you should study more than all the chapters except for the end chapters, and that's because it introduces a lot of topics that we then went on and talked about later, like Koch's postulate was first talked here, endospores was first talked here. I think the cell wall was first talked here also. Infectious diseases first talked here, pathogens first talked here. And then chapter 1 goes into the brief history of microbiology, and I just enjoy history. You should know about the importance of microbes on the planet Earth and to people. Like, for example, we needed microbes to take care of the sewage in our sewage treatment plants. You should know about the beneficial roles that microbes play on the planet, like breaking down environmental problems like this oil spill. You should know about the naming, the classifying of microorganisms started by Carolus Linnaeus, which I think your textbook calls Carl Linnaeus, or Carl Linnaeus, sorry. His real name, I think, was Carl Linnaeus, and he liked Latin so well, he changed his name to Carolus Linnaeus. Started the classification of all organisms. He broke things into animals and plants, which we no longer use. We do have animals and plants, but we classify them into the domains. And he also started a scientific name where every species has a genus name and a specific epithet for its scientific name. And this name system that he started is still in use today. We call it the scientific nomenclature or the binomial nomenclature, because every species has two names, like Homo sapiens. Did you know about the three domains and then the kingdoms after that? All right, so the brief history of microbiology, you do not need to know the dates except for about 3.5 billion years ago, we have the first ancestors of life on the planet, the ancestors of bacteria, the first life on Earth. We have fossil records of that. You should know in the history who discovered what? Like Janssen discovered the compound microscope, Robert Hooke reported the first living cells. You do not need to know the years that these people did that, but you should know who discovered what. You should know the history of microbiology was largely concerned with the debate over whether microbes or all organisms could spontaneously generate or all living organisms came from another living organism, called the theory of biogenesis. Everyone always agreed that a baby came from two parents, but they did state that other organisms could come from spontaneously generate. And Francesco Redi gave the first evidence that spontaneous generation did not occur with complex organisms, where he also gave the first controlled scientific experiment. So you should know this experiment because it's the first documented controlled scientific experiment that was written up, were 3 jars that were covered with a fine nesh or seal, had no maggots growing in the meat, and then 3 open jars with the meat had maggots in the meat, showing that the maggots did not spontaneously generate. After Francesco Redi's day, everyone agreed that complex life forms did not spontaneously generate, but people still argued that microbes could spontaneously generate. And that wasn't solved until Louis Pasteur disproved spontaneous generation with his swan neck experiment, where he put media in a glass container, bent the glass container that way, so it's open to the outside so the vital forces could get in it, and there was no spontaneous generation in the media here. You should also know about Anton Leeuwenhoek, the first person to describe microbes. And he even saw bacteria, cells so small that nobody else saw them for about a century. And that's because he was the best microscope builder at the time and best lens grinder too. You should know about the germ theory of disease. I guess you should know about Koch streaking for isolation on solid media. This allowed for the easy development of getting a pure culture. You simply streak out a culture on a solid media, get a simple isolated colony, streak it out again to get isolated colonies, and then take cells from a colony that has gone through 2 colony formation events. And then the chance of you getting a pure culture is very high, over 95%. Pasteur also showed alcoholic fermentation. So he showed that fermentation goes and started pasteurization, why it's called that, after Louis Pasteur. He also was the first person to go through and show how vaccines work and then the steps for making a vaccine. And he went on and made a few vaccines himself, such as the rabies vaccine. Joseph Lister used the first chemical disinfectant to prevent surgical wounds. What he used was phenol, a substance which is actually fairly, what's the word I'm looking for? Aggravating to the skin. It's not something you really want to get on your skin. It's also a carcinogen, and it's fairly unpleasant to breathe. But it was the best chemical disinfectant that he was able to find at his time, and he gave it the disinfectant properties of 1. Now we have disinfectants which are much stronger than 1. I think the strongest we have is about 1,000, and that's called the phenol coefficient. Robert Koch showed the first bacterium, Bacillus anthracis, that caused the first disease, which was anthrax. He actually showed that, and then later wrote up his steps for proving that Bacillus anthracis causes the disease anthrax, and that we call Koch's postulates. As you know, Edward Jenner came up with the first vaccine. During Edward Jenner's time and after that, nobody understood why his vaccine worked, so no one could make another vaccine. But his vaccine worked because he was using cowpox, a virus genetically similar to smallpox, so that when the patient became immune to cowpox, they had cross-reactivity to smallpox. And so when they're immune to cowpox, they were immune to smallpox. And it was actually Louis Pasteur who showed that relationship. Alexander Fleming is the first person to discover an antibiotic. He discovered the antibiotic penicillin. Excuse me, penicillin. Penicillin. Penicillin G from the Penicillium mold. I suppose you should know that sulfa drugs were the first antibiotic clinically available. So it was the second antibiotic discovered, and people were able to make it man-made. And so it was clinically available before penicillin G became clinically available. All right, I've already mentioned you should know about the microbial normal biota. Any questions about chapter 1? Any chapter questions about anything in general? I have not reviewed the lab, but you should go through each lab and know the major concepts of each lab. I won't ask more than one question in each lab, but you should also understand the unknown project. And I think I've got 2 or 3 questions, maybe 2 questions, where I'm giving you different characteristics of a cell and then giving you a key to identify the cell. Any question on any of that? All right.
What are the essay questions? I mean, what's involved in that? Well,
Let me see what I can tell you. There's an extra credit question that I remember, in which case you don't need to study for that. I know. I might have to log in online. I'm not finding the final, so I'm logging in to get the final. Fortunately, I didn't have to get the system to give me a phone call to verify my existence. Every time I use Zoom now, I have to use the phone to verify my existence. It's kind of annoying, and that was why I'm a little bit late on getting the class starting. At least I got the phone near me this time. Oh, where's the essay questions? Make sure you're not seeing this. Yep.
that'd be okay.
Yeah, I'm sure it would be. Oops, don't want to close the class here, but get to the fine. Oops. Oh, come on. Hit the wrong button there, so I couldn't see the questions. All right, some of the essay questions, I'm asking you to identify the bacteria, and I'm giving you a key. So that's the unknown project. There's at least 2 questions like that. And that makes sense because that would take time for you guys to do that. So that's why it's an essay type question. You're not timed on that one. And then I'm giving you a case study where you're looking at MRSA, and I'm asking you questions about the procedures that are in place. Will it stop the spread of MRSA among a particular population which is at greater risk of getting MRSA, and then a bunch of other people at less risk, but they're exposed to the other people? Actually, that's only one question. The other question is also MRSA, and this one is a MRSA kept happening to a woman, and the doctor was suspicious of it because the woman was not becoming, not her, but the MRSA was not becoming resistant to the treatment for the MRSA, which is multi-drug resistant Staphylococcus aureus, by the way. And there was one antibiotic he was able to treat it with, and she repeatedly was coming down with MRSA, and the doctor was becoming suspicious because she was repeatedly getting it, and the MRSA was not becoming resistant to the drug that he was treating her to. So he suspected that she was continually being re-exposed to a source of infection, and the source of infection was not being treated. And so he began studying her house and then discovered where she was getting the MRSA. And so you need to figure out how to get rid of MRSA in the house. Okay?
Thank you.
And that's about it. All right, any other questions? If not, that's it for the review. Good luck. I hope to begin grading your assignments today. I haven't got graded, it's the two big ones. I doubt I will get the grades up before some of you take the exam, but I'll try and get the grades up for at least one of them tomorrow. Maybe I'll work on the unknown report because that'll take me less time to get it graded. Any questions about anything?
I don't think so.
All right. I will see you. Good luck on the final.
Thank you. Bye.
Bye.